All right, now that you are familiar with just the structure of the peptidoglycan, we're going to look at where it is in terms of the overall structure of the bacterial cell, and we're going to compare the differences between the cell wall itself of gram-positive to gram-negative cells. So both gram-positive and gram-negative cells, so just a basic cell, is going to have its cell membrane. So the membrane is it's phospholipid bilayer. And it'll be made up of transmembrane proteins and peripheral proteins and all the other molecules you would find as you study just generic cell membrane structure. Okay, so if you want a refresher on that, go back and look at just the structure of the cell membrane, and it'll have the details there. So in general, we have that. That's just the cell membrane itself. Now, outside the cell membrane, we actually have the cell wall. So the cell wall is going to be made up of, if we remember, repeating units of NAM and NAG. M, G, M, G, M, G, M, G, like this. And over here, gram-negative bacteria, they're going to have peptidoglycan, NAM, NAG, and so on. So we're going to have, this is peptidoglycan. And if you're not familiar with the overall structure there, you can go back and look at the peptidoglycan little lecture that I have on showing you, what does N-acetylglucosamine look like? What does N-acetylmuramic acid look like? How are they put together? Then how are the chains connected with the peptide cross bridges? So look at all that to see the details. So overall, in general, the chains themselves pretty much the same, and the way they're connected are pretty much the same. The difference here is going to be the gram-positive bacteria are going to have just many of these chains, and I'm just going to try to just quickly do something like this just to give you the feel for it, where it's very thick. All right, so this peptidoglycan layer in the gram-positive bacteria is a very thick, multi-layer peptidoglycan. In gram-negative bacteria, it's going to be thin, very few layers. It could just be 1 to 3 layers, of peptidoglycan, so nothing much. So focus on the gram-positive here first, and then we'll move into what else. So what else does the gram-negative have? So the other thing is to add in here is there's a space here you can see between the cell membrane and the peptidoglycan layer, and this is called the periplasm. And it's going to be really important as we study later on and we look at how do bacteria do things like run the electron transport chain and find out where are the active transport pumps of the electron transport chain, where do they pump protons to, how does diffusion occur, and so this space will be yet an additional space that a bacterial cell has that's not inside the cell itself in the cytoplasm, but because the cell wall then creates a secondary barrier, this is yet another compartment that a bacterial cell technically has to use to store proteins and enzymes and other sorts of things and ions, and we'll find that a variety of uses for the periplasm. So membrane, very thick peptidoglycan, and then for the gram-positive cells, they're going to have things called lipotecoic acids. So these are called lipotecoic acids. So I'm not drawing the whole structure, the actual detailed structure of them there, but they are these molecules that run all the way from outside the peptidoglycan layer through the peptidoglycan, through the periplasm, and then anchor to the phospholipids of the cell membrane. That's what I call the lipoteichoic acids because they go all the way through and attach to the lipids. They provide extra rigidity and structure and also anchor the cell wall to the cell membrane, kind of connecting them together. They have additional roles in bonding with other cells, for pathogenic bacteria they use them. There's a variety of different uses for identity based on these teichoic acids. Lipoteichoic acids. And now there are an additional group called, here, just teichoic acids. Now they don't have the stability role, but they do have the binding and adhesion role and identity role. They are present, again, outside the cell, and they extend into the layer of peptidoglycan, but they do not connect all the way through to the periplasm and the cell wall. So we have teichoic acids, a unique type of molecule found just in gram-positive cells. We're not going to see these in gram-negative cells. So teichoic acids and lipoteichoic. And the lipoteichoic ones go all the way through and connect. That's the main structure, really. It's going to be pretty similar between all the different gram-positive bacteria. Different types of teichoic acids can give different identities, but the core structure will be largely the same. There are going to be some additional things we'll talk about that can happen also outside this layer, but that'll come next. We want to stick with cell wall. So gram-negative cell wall. This is where it gets kind of complicated and though interesting. I'm drawing here yet another phospholipid bilayer. So down here is our phospholipid bilayer. That's our cell membrane. And now we have a second one. So this is technically not the cell membrane. It's just a second phospholipid bilayer. So this is not the cytoplasm inside here. It's still periplasm. Okay, the periplasm. So we have a much bigger periplasm in the gram-negative bacteria. So we have a larger periplasm. A second phospholipid bilayer. Okay. And then attached out here to this second phospholipid bilayer are going to be molecules referred to with this abbreviation LPS. They are lipopolysaccharides. Sorry, running out of space. I'll rewrite it here. There, just barely made it. So some of these two words are big. It's hard to plan them out. All right, so these are also unique for gram-negative cells. Or again, the LPS. They're unique, they're different between different gram-negative bacteria. They're just like they described. There are sugars with lipid components. They're embedded in the cell, the outer cell membrane, the second cell membrane. So they have a lipid component and a sugar component. They are also going to be typically exotoxins, in many cases. And when someone becomes ill from a pathogenic gram-negative bacteria, one of the reasons may be because of the LPSs. The lipopolysaccharides themselves are providing a reaction to the organism's physiology. Right, so these LPS are unique to here. We're also going to see some other things too. So we're going to see the ability to bring things in here. To transport. So the other thing is to get into this cytoplasm now, whether if you're a nutrient molecule, for example, it's not, there is a large peptidoglycan over here, but it has properties that are more compatible with polar molecules. Here you have a second non-polar barrier, right? Which then makes it far more difficult than for a lot of the polar molecules, which are nutrients, to be taken up or transported by the cell. So we have to have additional transport proteins that are going to be found out here that are going to be able to get molecules then into either the periplasm, and then from the periplasm then actually into the cytoplasm of the cell. So it has to be sort of a second transport system found in the gram-negative bacteria. So that's something that the gram-positive bacteria typically don't have or don't require. That's the main breakdown of it. There are going to be some other fine details that we would get into later, but if we were to rattle off a couple of the specific differences between the two and similarities. The first similarity is cell membrane, periplasm, and then peptidoglycan, sort of in order. Now, starting there, we get the differences. Very thick peptidoglycan in the gram-positive, a very thin peptidoglycan in the gram-negative. The gram-positives have these long lipoteichoic acids and these short little teichoic acids, which are unique for the cells, and they're unique for gram-positive, and they provide a variety of different roles in identifying cells and binding with other cells. The gram-negative cells have a second phospholipid bilayer. They then have lipopolysaccharides embedded in that layer, and they have to have additional transport proteins that can then move molecules then in through. So they also have then a much bigger periplasm space. It's kind of broken up a little bit by the peptidoglycan. As we study then where does this name come from? So what does it mean, positive or negative? People start to think it has something to do with charge. It has to do with the type of stain that we discuss and how the stain can actually bind into these areas and then be washed away with alcohol or not. And in this particular area here, what we're going to find is the stain will be bound with a mordant, and then it won't be able to be washed out. In this area here, the alcohol is going to disrupt this outer phospholipid bilayer, and then the dye is just going to like go away, so it's going to lose the color, essentially. So one of the next things we'll look at is, what is the gram stain, and how is the gram stain used to determine which type of membrane that a cell has. If I give you a bacterial cell and say, is it a gram-positive or gram-negative cell, you couldn't tell just by looking at it. You'd have to stain it first and do a very specific type of stain, which is really a test or an assessment of the membrane structure itself. So be able to list these differences and similarities, be able to just do a quick little sketch with some labels, of the membrane, or identify structures from cell wall membrane that's already been drawn for you, and say which, is it gram-positive